Hai, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Adib Danial bin Burhanuddin. I'm from class 3C and my matrix number is 2022930925. And today I'm going to talk about vascular wilt disease, or in scientific name it is called Fusarium oxysporum F species elidis. So, a vascular wilt disease are caused by different fungi that attack the vascular system of trees. A tree responds by blocking its vascular system to contain the disease. In doing so, the water supply to the leaves is cut off. So, this is the example of the vascular wilt disease that attack the oil palm. In mature palms, the disease can manifest in various forms, which is in chronic form, acute form, and temporary wilt form. In chronic form, all the leaves dry out, the rachis breaks near or further along the base and fronds hangs downwards. As the disease progresses, younger leaves are successively affected, while the erect, younger green leaves in the crown shrinks in size and may become chlorotic. Palms can persist in this state for several years before the crown collapses. Acute form, leaves dry out and die while still upright and then snap off about 1 month or more from the trunk, usually during strong winds. Diseases progresses rapidly and palms die within 2 or 3 months. Lastly, temporary wilt, leaf systems develop but palm recover subsequently. On immature palms, leaf in the middle of the crown become yellow or brown. The discoloration spreads to the lower leaves and the palm eventually desiccates and die. At the nursery stage, infected palms show progressive shortening of younger leaves and desiccation and death of older leaves. These symptoms are thought to be caused by a combination of water, stress, and changes to plant gibberellin levels or activity. Internally, this disease is characterized by discoloration and blockage of xylem vessel with tolicles and gums. Vascular discoloration occurs in palm stems and can spread systemically to the petioles in severely infected palms. Roots do not exhibit signs of infection even in highly diseased field palms. Next is detection and inspection. Brown discoloration of xylem tissue in the trunk leaf bases of severely infected palms is diagnosis of disease. Care must be taken to distinguish between brown xylem vessels and discoloration of the sclerenchyma tissue of vascular bundles, which can occur in old healthy palms or following attack by ganoderma or armillaria. Trunk samples can be obtained by felling the palm or preferably with an increment borer, which avoids destructive sampling of entire palms. Samples can be examined microscopically for Fusarium hypae or vascular occlusion with gels, or more reliably, latent onto agar. The pathogen can also be isolated from seeds and pollens. Infection can be confirmed by surface sterilization of samples, then plating onto Fusarium selective medium. After 3 to 5 days, mycelium of F. oxysporum emerging initially from xylem vessel should be visible. F. oxysporum can be diagnosed to species level at reasonable speed by DNA-based diagnostic using PCR with primers based on the translation elongation factor gene. There is still a need to offer patotype-specific primers. Lastly, prevention and control. Phytosanitary. Almost all batches of seed may be contaminated by F. oxysporum. The import of germplasm material must be accompanied by an import permit issued by or on behalf of the Director General Department of Agriculture for Peninsular Malaysia or the Director Department of Agriculture for Sabah, and a phytosanitary certificate issued by an authorized official from the country of export. The import conditions are available upon request from the Plant Biosecurity Division Malaysia. All consignment are subjected to inspection by the Department of Agriculture prior to clearance by customs. Germplasm material imported from high-risk areas should be sent for third country quarantine before arrival onto Malaysian shores. The seed can be vacuum treated with a blend of fungicide, carbendazim, and prochloras administered under the pressure of 15,000 to about 8,000 PA with the aid of a wetting agent. cultural control and sanitary methods, the pathogen persists on sites of previously infected palms. The disease's incidence can be reduced by planting new palms more than 2 m from old stumps. The application of potassium and the removal of ground cover plant and plant debris mulch will also help in reducing diseases in incidence.